Good evening, everyone. My name is Hui. I'm working on a PhD project co-funded by Ghent University and the Flanders Research Institute for Agriculture, Fishery, and Food in Belgium. And I am one of the soil scientists here. And I will give a bit background. Crop residuals is the main sources for maintaining soil organic carbon and nutrient in agricultural soil. The removal of crop residuals could reduce the soil quality. On the other hand, they could be used as other alternative utilization, for example, fodder production and biofuel, which could bring some benefits. And biofuel is also one of the solutions for climate mitigation. So to be or not to be incorporated still remains a question. Today I'm going to talk about part of my project. We want to check whether above ground and below ground biomass had different short-term degradability under field conditions. Different biomass sources have different behavior in the soil. The previous review suggested root-derived carbon is 2.4 times more efficient than shoot-derived carbon in build-up and preserving soil organic matter in long-term experimental fields. There are some hypotheses proposed under estimation of root carbon. It is indeed very difficult to quantify the root execute date during the growing season. Biochemical composition. For example, root has more lignin than shoot. Physical protection via the soil aggregates. The carbon availability within the soil aggregates is limited for decomposition. And then we are focusing on the third mechanism. This is research done by Kohn and Six in 2010. They used C13, stable carbon isotope labeled shoot and root to see how they stabilize in the soil. So from the result, you can see the root-derived carbon, they found more root-derived carbon in the microaggregates than the shoot-derived carbon in 3 different crop systems and in different sampling events. So this close contact between soil and roots could explain the relative stability of roots. So our first hypothesis is root and shoot has different short-term degradability under field condition. If we could confirm that, and then we know when relatively more root is stabilized in the soil. Of course, scientists would like to understand better the mechanism, so they mimic the situation, how the stabilization in the lab, but it didn't work out. They found a similar contribution of roots-derived carbon as shoot-derived carbon, which is different from the results we get from the field experiments. So we think it might be the translocation and physical disturbance that's introduced in this lab-based incubation caused this different result between field and lab experiments. And we would like to test it under the field condition. How did we compare? So we incorporated maize root and shoot in light sandy loam soil during 2 seasons from 2016 to 2018. Each treatment has 3 replicates, and we have our pre-experiment root-exclusion approach in maize fields and main experiment natural C13 labeling approach in a C3 soil. And we also use different systems to detect the soil respiration. I will talk about the approach and the system in the coming slides. In the first season, the root exclusion approach, we chose a maize field, which has been planted maize for a few years. A maize plot with 4 square meters, we remove the roots, it's treatment 0, and the treatment with maize roots. The maize was taken out and chopped into small pieces and incorporated back to the top 30 centimeters. And the different soil respiration for these 2 treatments will be the decomposition of the roots we added here. So this is how we measure the CO2 flux in the field, an automatic soil CO2 flux, LICO 8100, equipped with a chamber. And there is an infrared detector in the box, so we could check how fast the CO2 is released from the field. And at the same time, we measure the temperature and the moisture at the same time. At the beginning of the experiment, the sensors were installed and temperature, moisture was recorded every hour during the whole experiment. We used the model of CO2 flux with a function of temperature, and we could know the flux during the whole experiment period. Here I'm showing some results, how the, here is a timing. We took the measurement every 3 weeks, and the y-axis is the CO2 flux, how fast the CO2 is released of the two treatments. You can see the temperature plays a very big role. In winter it's quite low, and in summer it's higher. With the, we know how fast the CO2 is released, and we could calculate during the period of October 2016 to May 2017 how much of carbon is released from these two treatments. So the first bar is without roots, second is with roots. As we mentioned, the difference between these two treatments is the decomposition of the roots that we added. And we know at the beginning how much carbon was added in each plot. Even though you see some difference between plots, in general, we see most of maize was decomposed after 8 months. And then we come to our main experiment, nature C13 labeling approach. This approach only works the first year transition from C3 crop to C4 crop. So we selected a C3 soil, which is a long-term grassland, with the delta C13 minus 26. And then we harvest from those 4 square meters maize plot. We chopped maize shoot and root into small pieces and incorporate it to the top 30 centimeters. And then we're going to compare the degradability and the field condition. This is another system, a closed chamber-based. Then we took gas samples every 15 minutes, and then we took this gas back to the lab, analyzed the CO2 concentration and the delta C13 of this CO2. With the build-up of CO2 concentration, we could know the CO2 flux, and with the delta C13, we could know how much of this CO2 was derived from maize. So this is the equation we applied. It's a bit complicated, but I will explain it. So we have the maize plant with delta C13 with minus 13 here, and we have the soil is minus 26. And then the more CO2 derived from the maize, the higher it gets close to the minus 13, so become more less negative, we say. Yes, so this is how we calculate the CO2 fraction. And this system was applied every month. And with the two treatments, this olive color is incorporated with roots, and the green color is with shoot. And you can see this is the maize fraction during the experiment period. Oh. And you can see in general, it increased and decreased a bit. Of course, the temperature also affects maize fraction. If we have a close look at the maize fraction in the treatment with roots, you can see after May, we can barely see maize-derived CO2, which was consistent with our previous pre-experiment that shows after 8 months, most of maize was decomposed or we say stabilized in the soil. And if you look at the shoot treatment, it still remains around 20%. But when you look at the carbon input of root and shoot at the beginning, you will not surprised because the shoot carbon input is more than 10 times than the root carbon input. So maybe the quantity matters. And we know how much CO2, how fast the CO2 release, and we know how much there from the maize. And then we can see how much of this carbon was decomposed during this period. But you see some unrealistic numbers. This overestimation could be caused by the low frequency of the measurement, because it's quite expensive to do the analysis. So in the next season, we would like to increase the measurement frequency and also check the effect of the quantity. So the second season, we started our Nature C13 labeling. Again, we incorporated the maize shoot and roots into C3 soil. We added one treatment. The same amount shoot carbon as root carbon was added, and we're going to check the effect of quantity. And we also use another system to cavity Ring-Down Spectroscopy. We measure it every 2 weeks. Be honest, it's not so fun to carry such huge boxes around the field. But the good thing is it could measure CO2 content and delta C13 at the same time in the field. And you can see the build-up of the CO2 over time, and the delta C13 decreased. This is the raw data from this machine. I just want to show you how it works. And again, we know the CO2 flux. We know the maize fraction. We could see this amount of maize was emitted from the period of October 2017 and to May 2018. We also know the amount of carbon input at the beginning, and we know such amount of maize carbon was decomposed. So actually, the shoot and root have similar degradability under the field condition, and we check again the normal amount of shoot, which is 3 times more than root carbon input at the beginning. But the effect of quantity is not significant. So let's summarize. After 8 months, most of the roots was decomposed under field condition, as we barely see the maize-derived CO2 released from the treatment with maize roots. And the short-term degradability of above-ground and below-ground biomass is similar after translocation. This did not support our first hypothesis that we think the short-term degradability will be different between root and shoots, but it did support our second hypothesis that translocation and strong physical disturbance that introduced in the lab incubation could be the reason explain why it's different in lab-based incubation and the field experiment. This experiment is still going on, so we are also going to check the root degradability without physical disturbance. If we could see that root degradability is lower without physical disturbance, then we could say that the physical protection actually play a major role for this relative stability of roots. And then we come back to the first question, to be or not to be incorporated. In the point of view of accumulation of soil organic matter, you see that roots is more efficient, but residual return does help to prevent soil erosion and also bring back other nutrients to the soil. How about we incorporate them partly, and the other part we use as a biofuel or fodder production. So on one hand, you could reduce CO2 emission by reducing the amount of decomposition of residual. On the other hand, you could get some economic and ecological benefit from it. Yeah, that's it. Thank you very much.